So, hello everyone. Hi, hello, and welcome again. Hi, Microbehunter here. I'm Oliver, and welcome to another Saturday Microscopy live stream. Today, a little bit more microbiology, but yeah, plenty of microscopy as well. Like every week, I would like to take the first couple of minutes to sort out all of the technology here. So I seem to be online. Chat seems to be working. I think the sound is also working. Yeah, and yeah, hi. Now we can actually really start. Today, luckily, I'm not quite pressed for time like I was last week. So we can take our time today a little bit. And today's topic I want to completely dedicate to bacteria. And yeah, as you have seen in the title, specifically safe bacteria, because working with bacteria and generally is a little bit or could be a little bit problematic, but not today, because the bacteria that I will be working with belong to biosafety level 1. So basically, these are bacteria that are harmless, yeah, and that's quite important. Okay, so this is basically the how do you say the idea for today, and what I will do as well is not only work with show you a little bit some techniques here with petri dishes, but also I'm going to use my centrifuge because I'm going to show you a short extraction technique from yogurt bacteria. So let's see if the other camera works. So let me see. Yeah, here it is. So that's basically the centrifuge view. Okay, I'm just checking whether this also works. Okay, so yeah, first 3 or 4 minutes, usually a hello around the world. I usually read the greetings from the chat because people need a little bit of time to join in. So hello from North Carolina, the United States, from Vietnam, yeah, from New Zealand, from Northern France, okay, England, Greece, England, Canada, from Austria, yeah, very nice, from Florida, and yeah, from Poland, from Serbia. It's very nice. So apparently a lot of people like microscopy. Yeah. So I need to explain a little bit of what we've actually got here. So first of all, just a short disclaimer, I do not recommend that if you basically are into hobby microscopy, I do not recommend that you grow your own bacteria. You got to be careful because strictly speaking, if you cultivate unknown bacteria, then you're in biosafety level 2. Level 1 is safe, and that's generally allowed, even at home. But biosafety level 2 already requires, in many countries, a specific laboratory license because you don't know what you're working with. And there are certain laws and regulations. Then there's also biosafety level 3, which is even more dangerous. Certain viruses like HIV belongs to that. And then there's also biosafety level level 4, where you have to wear a spacesuit when you actually work, with those viruses and bacteria. And the thing is the following, that I'm going to today only work with biosafety level 1, and this basically means the bacteria that I have here, over here, E. coli bacteria, I got from basically from capsules that can be eaten. They are there to regenerate the digestive system. And so they're basically safe. It's already expired, but it still worked. And the fungi and bacteria that I have over here, it looks kind of dangerous, all of the mold here, but that is actually something that I isolated from cheese. Okay, so this is actually also edible, and I have subcultivated them, and I'm going to demonstrate a little bit of how I've done that, and of course we're going to put them under the microscope. Yes, Ever Small already wrote a comment, because just before the live stream I checked, did I find the reflected light pictures? Please, can you resend them? I could not find them in my email, but thank you for the, I didn't forget about it, but I honestly could not find them, and I checked the emails, so maybe, I don't know, maybe I overlooked them or they got lost. Maybe, if you could send them again, would be really great. Thank you. Okay, so just a little bit comment here. So basically, what I've got here is those from cheese, so they're edible. The fuzzy thing here, that is a fungus. and those colonies over here are from bacteria, most likely Brevibacterium. So a little bit later, I'll show you this a little bit later, and I'm also going to show you how I'm going to subculture them in a fresh Petri dish. This Petri dish I made myself. Doesn't work quite well. The growth medium is not ideal. The other Petri dishes, yeah, I did it the, how do you say, the expensive way. I ordered them ready-made from Amazon. They have different types, yeah, pretty expensive. Yeah, but it's possible to actually order them ready-made as well, and there are different types. And actually, these are designed to be for fungi, but they also work for growing bacteria. Okay. This is a Pringles Petri dish. Are you referring to the lid of a Pringles potato chip? Yeah. No, it's actually, these are the standard these are standard plastic Petri dishes, which basically can be also bought. Okay, so but what I want to do now is the following: is I'm going to show you, before we do that, I want to actually show you, and I tried this today and I was quite happy with the result. I'm going to now show you how you can extract yogurt bacteria from commercial yogurt. Now, of course, you can try to directly put it under the microscope. However, it doesn't work. It doesn't work that well. Okay, and there are a couple of questions here as well from Tini Coco from unwanted bacteria. I will talk about the aseptic technique later on when I talk about the Petri dishes. And if you put the yogurt directly under the microscope, it's gonna, yeah, you can see bacteria difficult because there is so much milk, protein, and fat and so on present from the milk as well, that the bacteria are not very concentrated. So I'm going to show you now a way on how you can concentrate the yogurt bacteria. Yeah, you know that I'm a teacher, and just yesterday, during my biology lesson, I talked about bacteria, and I told my students that there are, of course, you have bacteria and fungi on cheese, and also yogurt. Yogurt is almost 100% lots of bacteria. Of course, many didn't know that. They were quite surprised that when you eat actually yogurt, you're eating mostly bacteria, right? And I would like to show them to you because they look kind of nice. But let's do a little bit of extraction first. So first of all, I want to separate the bacteria from the rest of the coagulated milk and the protein and so on. And in order to do that, I have to centrifuge it. But this yogurt here is way too thick. Yeah, so it's not suitable for that purpose. And what I have to do now is I have to dilute it down with a little bit of water. And I don't know. I'm just gonna add maybe approximately the same amount of water, okay, to the yogurt here. And I just realized that this takes a little bit of time. Okay. Yeah. So this is completely, honestly, completely arbitrary right now, right? Maybe the same amount or double the amount. Yeah. In any case, I want to have a very liquid suspension here. Yeah. It's almost like milk, really. Okay. And while I'm doing that, I'm reading the I'm going to be reading the comments here. Yeah. Okay. Basically, the process of extracting lactose from milk. Heaven's sake, I don't know how to do that. Thank you for the suggestion, but I don't. But you see, even if I don't know how to do that, this is actually something to dig in and dive in a little bit and try to figure out how you can do that. Okay, yeah. I know that it's possible to actually measure the sugar concentration in a variety of ways, but to actually extract it and to crystallize it, that's a challenge. Okay, so let's have a look at some other comments here. Yeah, how do we keep the Petri dishes clean from unwanted bacteria? It's the aseptic technique. I will talk about that, right? And yeah, so yes, if you have any comments or questions to me, please feel free to post them and write down at Microbehunter or at Oliver, and any off-topic questions I will try to answer later. I still do think that this is a little bit thick. Okay, so I'll add a little bit more. Okay, and what I'm going to do now is the following: I'm going to put this, some of this into a centrifuge tube. yeah, plastic tube, and I'm going to centrifuge it for, I don't know, maybe 20 seconds at a very low speed, so that all of the high density, basically coagulated milk and so on, is going to separate. Okay, so and I somehow have to get it into this tube here, and yeah, and this is going to be it's going to be a little bit of a mess, I suppose. It actually works. Okay. So it's like milk. Okay. That's what I feared. Not to worry. That's why you always keep some tissue paper close by. And when you want to centrifuge, you need, of course, a counterweight. In order to make sure that it's balanced. Yeah. So that's basically the first thing here, and I need a counterweight, and you generally want to make sure that it's more or less the same. You see that over here? That's just a tube with regular water, right? And you see that there is a little bit more water in here than here. So what I'm going to do is I'm just going to add a little bit more water here, and fill it up, because if I don't do that, then the centrifuge might start vibrating, and this is also then very noisy. In the past, I have used a scale to really make sure that the mass is the same, but I found out it's not necessary. So that's the yogurt water, and this is the counterbalance. Now I'm going to switch over to the centrifuge, and I'm going to now really spin it down using maybe for about 20 seconds maybe. Here is the other one. There's a lid here. It's going to be a little bit noisy now, and this should be enough for the first step. Okay, so I'm just going to twist. So that's the lowest. That's the lowest setting. Okay. Yeah, it's kind of vibrating quite a bit. Yeah. I have this. It says here I have the same centrifuge. It's the cheapest one that I got, but it actually works surprisingly well for my purposes. I don't know, it was actually quite ridiculously cheap, and the reason why I bought it also is because if I collect water samples and I want to concentrate the water organisms for microscopy, then this also works. Yeah. Okay. So there's a suggestion, put it on a non-skid trivet. Okay, so there are actually suction knobs on the bottom, but because the table is not entirely smooth there, yeah. So I think that's going to be enough. So let's see if this was actually indeed enough. I tried this out before the live stream, and it actually did work quite well. So I hope that I'm able to repeat. And yes, indeed. Okay, so I'm going to switch again over. You cannot see it here. But let's move again over to the desk view. So, what you have here is you see that this one is quite cloudy, right? And down here it's solid. So what I'm going to do is I'm just going to throw this away here, and the bacteria are still up here. Yeah, and I'm going to do this a second time. I'm just going to take this, the supernatant as it's called, and I'm going to now do this a second time to remove again more of the solid yogurt material, which I don't need. Yeah? So I'm going to do this the very easy way. No, that's too risky. I wanted to pour it over, but there's always the danger if you start pouring it, then also the pellet, as this is called, will also start to come off. So what I'm going to do now is I'm just going to use a pipette. Okay, and I need a second tube. Just going to use this here. Okay. Let's move this a little bit out of the way here. So this is a little bit the safer way to up. I've just realized that the tip is not. So now. Okay, now it's on. Okay. Yes. So the bacteria, they will not sediment down because they are not dense enough. And I'm going to spin it again at a low speed. So that's basically the, how do you say, the demonstration on how you can, because without cultivating the bacteria yourself, you're still able to see them. So this is, I think that's enough. I'm just going to leave this in here. Yeah, this can be thrown away. I'm going to wash out the tube later. And what I'm going to do now is I'm going to now do this a second time to again remove anything. I'm going to do it another time. And let me do that here. So I have to now remove some of the liquid over here because it's too much, right? So I have a trash. This is my trash container. Whoop, I bumped it into the camera. So what I'm going to do is I'm just going to estimate it. Yeah, I got it. I got it pretty much. Okay, fine. Yeah, almost. Here it is. Yeah, it's pretty much the same. So, and again, this is the second time, the same thing. Yeah. So, and where is here? Let me see. Yeah. So that's one of them, and that's the other one. Again, at a low speed for a couple of seconds. This will again remove the remaining. Very loud. The remaining yogurt particles here. Okay. I'm just reading. I'm just using the time to read the comments here. Watching this makes me realize I should design a second version of my custom tube rack, one that holds the tubes at an angle so you can see what you're Yes, yeah, I'll show you what I've got here. That's the rack. That's a commercial rack. Yeah, but indeed it would be. Look, I have also another rack over here for big tubes. I already made a video about this some time ago. But for the smaller ones, actually, indeed, that would be also quite useful to have. Yeah? So I think that's also going to be enough now. See that the whole centrifuge is kind of moving around here. Okay. So again, let's go. It still was a little bit vibrating, so it was not properly balanced, but it's okay. Yeah, so that's the nothing in here. That's just the how do you call this? The water, the counterweight, and here, no. That's interesting. Okay. Got it. That's okay. Wasn't even necessary. I just realized. Maybe. Okay, I'm going to show you. This actually worked better than I thought. Because, look, it looks pretty clear. And then there is, over here on the side, the pellet. And honestly, I think this is already... And the water looks already pretty clear. So what I'm going to do now is the following. Should we give it a try? I wonder if... You know what? Actually, I wanted to spin it at a much faster speed now. But you know what? I'm going to do the following. I'm going to simply take this here. I'm thinking because the water is already so clear that there are almost no bacteria in here, so they might already be compacted down here. So you know what I'm going to do? I'm just going to take the risk now, and I'm just going to take this pellet here and put that under the microscope. Yeah, because maybe this was already enough. I'm thinking, no. You know what I'm doing? I'm going to spin it again, but I'm going to spin it at a much higher speed. Okay, that's the one. I have to make sure that this one again, because then maybe any remaining bacteria that I have to make sure that this one is pointing outwards. This one over here, the pellet has to point outwards. And this one is the counterweight. And now I'm actually going to spin it again, and I'm going to go up with the speed. So... I actually, why am I doing it like this? When I tried it before, there was still so much yogurt material, milk, protein, and coagulated fat in there. So actually, this looked actually much cleaner right now. So I'm going to spin it at a higher rate to really get all of the bacteria out. Yeah. So, and slow it down a little bit. So that's the water. Here we go. Okay. So, actually this looks not much cleaner right now, and maybe the desk view, maybe this actually is going to work. You can see over here. Need to focus. Yeah. Of course, there's still going to be some yogurt in here, but there will be also more bacteria now. And the water, the supernatant, looks fairly clear. So I hope that there are a few bacteria in here. And we're going to now put this down here under the microscope. Yeah. So I have to now remove and throw away the liquid. Okay. I'm just also checking the time a little bit. This actually takes a little bit longer than I anticipated because the main part should actually be working with the Petri dishes. And I'm also still gonna give it some time, but just as a little demonstration. So, and what you want to make sure is you want to make sure that you don't take out all of the liquid, but there should be a small amount left because you're going to use that now to resuspend everything. Okay. And we're ready to do a little bit of microscopy now. Okay. So, we take a slide. Here it is. We put this away so that it doesn't tip over. And a really small drop now goes on here. So if you want to observe bacteria, you have to really make sure that the amount of liquid is very small, very, very little. The cover glass should not float. So I hope this is one. And I drop it on here, and that seems to be fine. And now we have a look under the microscope. Okay, there's some water here as well. Okay, and let's see if we're able to see some of these bacteria. Let me check. This is the scope view. Yep. Okay. All of those larger clusters that you see are sometimes also, yeah, it's coagulated milk protein, yeah, but let's turn it up, and we go up yet more. Yes, it's kind of spreading now a little bit, the capillary action, but maybe you can already see, so that 60 times maybe you can already see the bacteria. So, yeah, unfortunately, it's still starting to stabilize, so that's why there's still some movement. But actually, I'm quite happy with the result, but I might have to explain it to you. But I kind of hope that the whole thing starts to stabilize. Yeah, but yeah, we're just going to wait. So first of all, here's the arrow. Those clusters that you see here, the stationary ones, that's still a lot of the yogurt, right? Coagulated milk and so on. But the things that you see floating between here, these are all bacteria. And you can now see them much better. If you put yogurt directly under the microscope, then you won't be able to see them as well because there is going to be so much of this yogurt protein and casein and I don't know what it's called, right? That you cannot see the individual bacteria. But right now you can actually see that the bacteria are long ones, rods. There are also small ones in chains. Okay, and so yeah, for example, those here, right? The little cocci that are in chains, and they basically are usually a combination of lactobacillus, and also Streptococcus and so on, which basically ferment the milk. So over here, for example, quite nicely visible, yeah. They're in tiny little chains, and that's something that essentially is, I think, a nice demonstration object. I mean, for example, me being a teacher in school, of course, I want to make sure that students they don't have to play around with dangerous bacteria, but yogurt itself is quite nicely observable this way. So I'm going to switch over to phase contrast now. Okay, here we go. Yeah, so this is now 40 times, so it's the magnification is not quite as high. You see that all of this milk protein is white, but the bacteria appear to be dark, yeah, here in the background. Yeah. And yeah, so this is actually one of the advantages of phase contrast, that you're able to see those transparent bacteria quite well. Yeah. The clear liquid is also possible. So especially if you make yogurt yourself, and then there will be sometimes this clear liquid on the surface. Sometimes in commercial yogurt it's already mixed in, so you don't have it separated. And for this reason it's a little bit more difficult, and that's why I've centrifuged. But over here it's, yeah, you can actually see the bacteria quite well. Yeah. So I just wanted to quickly show that to you, but I think maybe you're probably more interested, probably also in the bacteria that have grown on the Petri dish. So here again, it's 60 times. Yeah. And now it's a little bit, everything starts to stabilize a little bit. You can actually see over here, yeah, those little cocci which are in chains. Yeah, you've got some rods which are long. So that's the long ones are probably lactobacillus. Yeah, and you basically have yeah, different types of yogurts have their own different types of bacteria combinations here. Okay. There's a question: what degrees do you hold basically, I've got a master's degree in microbiology, and I've got also a second master's degree in computer science and biology education. Yeah? So that's basically why I've during my master's thesis at university. I've done, I don't know, a lot of these things here. Yeah. So DNA analysis was probably one of the things that I liked doing most, but lots of analytical chemistry as well, and so on. Yeah. And then I chose to change jobs, and now I'm decided to go into education. Yeah. In what's my bachelor's in the university, it's also biology. Yeah. So basically, I've got basically at our university, we just kind of skipped the bachelor's and made directly transferred from completed the master's degree. Yeah. So this is because I went to university in Austria. At that time, when I was at university, they did not have a bachelor's master's distinction, but you made the master's degree right away. Yeah. So. Let's basically, yeah, now it's starting to a little bit stabilize a little bit, and now we can see it actually much better. Yeah. So what I want to do now is I want to, I want to, the clear, yes, if you actually observe the clear liquid, then this is also going to work. Yeah. Yes, bright field. Yes, thank you for the recommendation. So 3, 2, 1, that's bright field. Too bright, I have to go down with the light intensity. Okay, and that is now regular bright field. So that's actually the way that you would be able to, yeah, not quite as colorful, yeah, but that is regular bright field. Yeah. At this magnification, unfortunately, I do not have dark field, so I'm not able to show that to you. Yeah, but that is bright field, and we see actually it's still quite nicely visible. Yeah, you can actually also try to play a little bit with the condenser. Yeah, that's too dark. So, okay. Yeah, so I'm going to now move on a little bit, and I'm going to now show you basically, some of the bacteria that have grown here, and I'm also going to show you how you're actually going to do plating. Yeah, okay. So that's, basically the next part here. I'm a teacher in high school, middle and high school. I was actually also teaching at university some time ago, but not anymore. So I teach biology right now, even though and in the past I also used to teach chemistry and also computer science. But there's too much going on, so I decided, okay, I'll basically focus primarily on biology now. So what I'm going to do now is the following. I'm going to quickly show you and I'm going to explain to you, assuming that you don't know anything about this here, I'm going to explain to you what you're able to see here. Okay? So maybe some of you already know this, then please bear with me. Okay? But what you have over here on this agar plate here on the side, this is a growth medium. Okay, which contains yeast extract, nutrients, they're different, I don't know, maybe hundreds of different recipes. And it's like a gel-like substance, but it's not gelatin. It's called agar. It's a polysaccharide. And essentially, when you now put bacteria on the surface here, then they will start to grow, and the bacteria will form tiny little dots, so-called colonies, which you're able to see over here not so well because they're too small, but over here you're able to see them quite well. So that's the one from the cheese. Okay, and you see that each one of those tiny little dots here contains millions of bacteria, and they all come from the same bacterium because it divided 1, 2, divided 4, 8, exponential growth, until you're able to see the colony. So so basically each colony over here comes from one bacterium, and therefore you can actually know where, on the original plate, where there was a bacterium. Right? So actually you do that to determine the so-called the viable count. If you put a water sample and you plate this on the, and then after a day or two you count the colonies, you know how many bacteria there were in the water sample. Yeah, so you can actually make measurements, and you can also see that some of the colonies, they have different sizes, so they're probably from different bacteria. And this fuzzy stuff here, well, that is a fungus. So it actually shows that the surface of a fungus was actually not a pure culture, but actually a mix of different fungi and bacteria. I can actually just see it from here. Yeah. What I've done is I've taken yesterday one of the bacterial colonies, and I spread it on this plate over here. So this is actually 1 day old, yeah? And you can see that it actually looks much more uniform and homogeneous. I'll explain to you what I've done here, why I've used this pattern. But what I've done is I've actually made a little video over 3 hours, a time-lapse video of this plate here, where you can actually see the growth of the colonies. Now, you're not able to see the colonies here because they're so small. So I put it under the plate under my stereo microscope, and I just want to show you how this looks like. Yeah, so these dots that you see here, these are now the colonies. And you know what? I'm going to speed it up a little bit. You see? And I'm going forward and backward, right? So you see, they're kind of growing. The colonies become larger, right? I'm going to go back again. Yeah. Yeah, so this is actually something that I've recorded over the course over the course of 3 hours. And so these are basically, pretty much, you see that, excuse me, they have pretty much the same consistency and the same color. So because they're all from the same original colony. Yeah. So the question is the following here: where did the fungi come from? Isn't the one you showed us the yogurt petri dish? No, this petri dish that I showed you here is I don't know, can you see it? Yeah, the one with the fungus that I just showed you, that is actually from cheese. So I scratched something off from the surface of a cheese. Yeah? Because again, as I said, I want to work with safe microorganisms. And the company that made the cheese sprayed the cheese with a certain culture of bacteria and fungi to give the cheese its specific flavor. Okay. Am I involved in any academic research? No academic research. Right now I'm basically decided to switch over into education. And that's basically what I'm doing here as well. And I'm not, of course, as a teacher. And then I, over this YouTube channel, I simply also want to share my fascination, also the hobby, so to say, with a wider audience. And that's basically why we're here. So, but no research. Okay, I don't have neither the equipment nor the lab or anything like that. So I would like to quickly show you now on how to do the following. What I would like to do is I would like to subculture or subcultivate those bacteria again on a fresh plate. Yeah. So which ones do I have? I'm going to use this here. Over here I've also got E. coli. And we're just going to put this under the microscope just later. But I just want to show you how this is actually done. Okay, how do you actually get bacteria over here on a fresh plate? I will tell you the following right now of what you should not do, and I hope I'm not giving anyone here any, how do you say, I'm not tipping off anyone here right now, but I'm gonna say it in anyway because on YouTube you find occasionally some folks who are, and also unfortunately this has also been done in schools, and it's actually not a good idea. If I now were to take my thumb and kind of press it on the agar plate, then essentially, if I were to wait then a day or two, I would actually see colonies of bacteria that were growing on my thumb. Right? You should not do that because what you're growing on here is then you're growing unknown bacteria which have the potential to also grow on your body and to be potentially infective. You're growing it at a very high concentration, number one, so you don't want to do that. So it's also a legal issue. And number two, maybe there are contaminants on here, microcolonies that I don't see. And if I basically press my thumb now on the plate, then I'm also transferring some of those unknown bacteria on my skin. And if I have a little wound somewhere, I don't know, yeah, you don't want that, right? So generally, I'm just saying you should not be doing that. And I don't. But this is unfortunately something that's when you watch YouTube and so on, you see this as a nice demonstration, educational demonstration, and then you wash your hands, and then the students roll again. They're washed. Some on here. No, don't do that. Okay, I'm just saying. Yeah, it's elevated biosafety level. So what I'm going. No, I forgot something. What I have to do now is there was the question: how do you actually prevent essentially bacteria from going in here? And that's indeed important. What you need is a gas burner, which I don't have and I'm not going to use because otherwise I'm going to burn the camera, right? But you need a gas burner for the reason that you need to disinfect the so-called inoculation loop. And also the gas burner creates an airstream of hot air upwards, which prevents bacteria which are in the air from falling down. Right, so you're creating, by the heat, you're creating this upward air current, and then basically you can open those plates without bacteria falling into those plates. Okay, so that's the thing, I cannot do that with a gas burner, so I'm going to use an alcohol burner, which is far less effective. Okay, because you don't have this intense airflow. I have to move the camera up, otherwise I'm going to burn the camera. Okay. And what I'm going to do, and I'm going to explain to you now what I'm doing. I'm going to first hold the inoculation loop into the flame to glow it out, so to kill all of the bacteria that are on here. I will then take a small sample from here. And what I'm going to do is I'm going to spread the sample on the side over here, and I'm going to just smear it on the medium. I'm going to disinfect it again, and I'm going to take a small sample from there again, and I'm going to dilute it again. And then I keep on repeating this. So every step here, it will become more and more diluted. And this is also what I have done over here. Yeah, it's very concentrated here. Then I've taken a few bacteria and I spread it over a larger surface, and I have done it again over here. Okay, so this basically means that over here I should be able to see individual colonies. Yeah, so a lot here, you take a small amount, you spread it, you take again a small amount and you spread it. Yeah, and that's what I would like to do over here, and then I have to wait a few days, and then hopefully it's going to grow. Yeah. Okay, so again I have to be careful that the camera. So I'm going to until it glows. Again, normally you would be using a how do you call this, a gas burner, which is more efficient, effective. See, it's pretty glowing. So, and I cannot pick it up yet because it's too hot. So I'm going to open this up now. I cool it here on the side. Okay, I'm cooling the loop. Okay, now it's cold. I'm taking a small sample. Okay. And I go over here. I don't want, and I'm now spreading it very lightly over here. Just the weight of the loop. Okay. And I again, kill off all of the bacteria. I have to remember where I spread it. I open it. I cool it. I'm now going to take a few bacteria from the original place and I will spread them over here. Okay. Again, I kill off everything that's on the loop. I cool it again. I have to remember where, yeah, I spread. I will take again a small amount here. And I spread it again. And I'm finished. Okay. So I'm going to now do the following. I'm quickly going to, yeah, turn it off. So what I'm going to, yeah, and now basically what I have to do is I have to seal it off. And I have to label it, and then I have to wait a few days, and I hope that this homemade medium actually works. It did not work very well with the E. coli. And this, I think, here is the greatest invention for laboratory invention. It's called parafilm. This is a really, I don't know, it's really nice, yeah, nice to use. It's very, yeah, versatile. I have to remove the paper cover here on the back. Okay. And yeah, I have to remove this here. Okay, and this here, yeah, is sticky. So I'm gonna go make it bigger here because yeah, yeah. How does parafilm look like fully stretched? I can try that. So look what I'm doing. I hold it here. I stretch it, and when you stretch it, it becomes sticky. Okay. Up. This was a little too stretched, a little too much. Okay. Okay, so I have to stretch it carefully, and as I stretch it, it becomes sticky. Yeah, this was a little bit too short. Okay, so I'm going to use another one as well. Okay. Because otherwise, it's not only to keep the lid on the plate, but also to prevent evaporation. Okay. And, yes, what do? You label it. Today is the 27th of September. These are basically bacteria from cheese. From cheese. Okay, and you store it like this with the medium, yeah, on top. Okay, so that's basically we're finished. Okay, so now I have to wait basically a couple of days, and then I have to hope that yeah, they will start to grow. And what I'm going to do now is I'm going to also show you how this one now looks. Yeah, under the stereo microscope. Where is this? The stereoscope. Need to turn on the light. Okay. Here it is. I need to, where is this? Here we go. I need to focus. Yeah, and here you can actually see, yeah, the tiny colonies. Let's have a look here. You see over here, this is where it was very dense. Yeah. Then I've taken a small sample over here and spread it apart. Yeah, and then I've taken again, and you see that over here it becomes less dense and less dense and less dense until you're able to see the individual colonies. Yeah. So this is yeah, it's what you call dilution plating. It's called. Yeah. So very dense over here, and then you always basically take a small sample and you start spreading it over a larger area, and then you see that in here there were the individual bacteria, and each one formed now a separate colony. Yeah. So that's basically the thing, and what I'm going to do now is I want to actually have a look yeah, under the microscope. So let me quickly check if there are over here, why only 2 spreads? Because generally it's enough. I mean, I'm not saying, so basically why only 2 of them? You can do more. It depends a little bit on the concentration of the original sample. Generally, it also depends on how many bacteria you're taking away here. So it actually works, yeah. But there are different patterns that you can use. Different people like different patterns. What I have done, I remember in the lab, I was so practiced, this was many years ago, I've actually only spread it over here once, then I've taken one and I was able to completely spread it a second time over here. So it was actually possible to do it in just one step. And I got individual colonies down here. Yeah? So it's a little bit a question of practice and trial and error. Yeah? So now what? Let's have a look at this under the microscope. And here we have to be a little bit aware that the bacterial density is incredibly high. Okay, so what I need to do is I need to take a very small amount of water as well, and an even smaller amount of those bacteria. And that's going to be a little bit of a challenge because, so I'm going to take here a little bit of water. And let's have a look at those bacteria which I have from cheese. Yeah, and I'm going to again, I don't want to contaminate this, so I'm going to do this again with a flame. Okay, so you can see it here a little bit. Okay, and I'm going to now take a small amount. Okay, here it is. Ah, yeah, I see there's this lamp here. Let's turn off the lamp. Yeah, over here. Oh, you don't see it. I'm glowing this here on the bottom. Yeah. So, and here again, I'm cooling it. I take a very small amount, and it's probably already too much because I see that it's cloudy. Yeah. So it's also finished now. Put the lid on top, and now basically what I'm going to do now is the following. I'm going to take a little bit of this and I'm going to put it over here. On here because I would like to dry it as well. I'm going to air dry this because maybe I can stain it later. Okay, and this one I'm just going to directly observe by putting a cover glass on top. Okay, and let's put it under the microscope. And I have to be honest with you, I did not try this bacterium yet. So let's have a look. Where is the scope view, microscope view, and let's see if I got the concentration right. Well, there's lots of stuff on here. What in the world? But actually, it looks, yeah, these are huge, these bacteria. Look at this. Actually, I mean, these are gigantic. I'm a little bit at a loss because they're so big. Could this be that this is some kind of a yeast and not a bacterium? These are not bacteria. They're way too big in my view. Okay, I learned something here. Yeah, this looks more like some kind of a yeast and not some bacterium. Okay. Why do I think so? Okay, honestly, yeah, No, these are not bacteria, in my view. The reason is if you look into the cells here, you actually see that they're not homogeneous. There seems to be some stuff. First of all, they're huge, right? And you actually see that there's something inside the cell, and these could indeed be cell organelles. So then this is a eukaryote. And sometimes you see over here a little bit. There's a large one and a small one next to each other. It looks like budding, and over here as well. Honestly, okay, interesting. Okay, my bad. I wanted to show you bacteria, and I think I ended up showing you some kind of a fungus, a yeast, I guess. But at least, well, still, okay. Let's do the following. Let's try phase contrast. Okay, and here we go. Yeah, this is, look, bacteria normally, bacteria normally look dark. This is way too thick and too large. Yeah. Okay, well, I think, yeah, I've also learned something. Yeah, not that I'm disappointed, but this is actually not, I'm almost sure that these are not bacteria. Okay, we've also learned something, but I do have some E. coli, yeah? Yeah. So this is quite interesting. Shouldn't be surprising, I mean, because of course the surface of a cheese is a complex mix, yeah, of different microorganisms. Yeah. Yeah. But the idea is, yeah. Yeah, but I think one of the things that you do see here, and that is indeed different from the actual yogurt observation that we have just done, is you see that there are basically no other, it's just cells, right? And so like in the yogurt before, I had all of this coagulated stuff as well. This is basically a fiber, because I probably from the tissue paper, because I washed and cleaned the microscope slide. So this is the reason why there seems to be maybe still from the tissue paper the fiber here. Yeah. Okay, so but in any case, yeah, that's basically how, yeah, one of the things from the, how do you say, from the cheese looks like. And what I'm going to do now is now actually I do want to show you some real bacteria, because I've got E. coli here. Okay. And so this one I'm going to put away. And where is the desk view again? 100 times oil for yeast. You know, okay. Oil immersion. You want to see it in oil immersion? You want to see it in oil immersion? Okay. Let's do that. Why not? Again, here's the scope view. So I'm going to show you how this is done. Yeah, you've probably seen this already before anyway, but let's do the following. Let's really go up with the magnification then. And I'm going to try to find a place. I'm going to try to find a place where the density is not too high. So maybe over here somewhere. And I'm going to remove, I want to get the arrow out of the way. So, and now I have to use my immersion oil, and let's give it a try. Let's completely remove the arrow. So, yeah, this, look, you can actually see that there is some kind of structure inside of the cells. Yeah. So let's do the following. Now you have to see a little bit what I'm doing up there. I'm going to first try to focus more or less with. Here we go. It's without oil. Okay, you see it's a little bit blurry. I have to engage the focus lock lever, which you cannot see, but that's what I'm doing right now. Now I'm lowering using the coarse focus, so it's kind of remembered now the position. The microscope stage is now the position is now remembered. I'm going to use the coarse focus to quickly lower everything completely. I'm going to now, let's turn down the light a little bit. So, and now I'm going to add the immersion oil. And now I'm going to use the coarse focus knob again to raise it up all the way. And because it remembered the position, yeah, it should be actually, here we go, in focus. Yes. Yeah, these are not bacteria. Okay. So, as a matter of fact, when you take yeast, baker's yeast, then it looks very similar. Yeah. And yeah, so that's basically now using oil immersion. So that's the maximum magnification that we can get. And yes, and we can now see that sometimes we have this, here. It's what you called it. That gives it away now. So I'm going to engage the arrow again. if you look very carefully, do you look at this cell here? That there seems to be this tiny little extension over here that's called a bud, or also over here, a bud. So that's an asymmetric cell division, asymmetric mitosis, also over here. And that means it's yeast. Yeah, over here, also over here maybe. Yeah, it's this asymmetric cell division. And yeast is a fungus, so it's not, yeah, it's, and also you can see that inside the cell, there is something visible inside the cell as well, some cell organelles, and bacteria don't have those. Yeah, also here and here. Yeah, yeah, it looks like a budding yeast. Yeah, and again, for those of you who joined in, I've got this from the surface of a cheese. Yeah. It's turtles all the way down. Haha. Who said that? Stephen Hawking. In the book, A Short History of Time. Yes. I can recommend that book. Okay, so what I'm gonna do now is I'm quickly checking the time a little bit, and I'm going to now put some E. coli under the microscope. Okay, so I'm going to remove this here again, and I'm going to put this all the way down. Let's remove this with the immersion. Well, I'm going to clean it later on. Okay, and let's go back to the desk view. Here we go. Yeah, and here we are again. And let's do the whole thing again. And now with, yeah, not this one over here. Look, I miss bacteria from cheese. This is how I labeled it. Not bacteria. Yeah, it's most likely some kind of a yeast. Okay. I mean, there are many different types of yeasts out there, so I'm not saying it's baker's yeast. So let's do the following. Now let's try it with. Now let's try it with. Again, I have to be a little careful with the how do you say, the camera, because I don't want it to become too hot. And so a little bit of water again. Okay, and E. coli. So that now is E. coli. How do I know? Because essentially I took it from those capsules, E. coli tablets. These are freeze-dried E. coli bacteria, which can be eaten so that you can regenerate your digestive system. So, for example, if you've been on a so-called an antibiotic treatment, and when you're basically the bacteria in your intestine have been killed off as well as a side effect, you can regenerate them by eating those bacteria. It's kind of, yeah, probiotic bacteria. Yeah. Why do you think you got yeast and not bacteria? Perhaps the cheese didn't have bacteria in the first place. A good point. Possible, but unlikely. The reason is the following. Look, I'm going to put if you look over here at the plate, you're going to see that there are different colonies. We've got definitely a fungus over here, and colonies over here, and they're not the same. So I'm quite sure that some of them might actually be bacteria and some might be yeasts. So it's probably a mix of both, but just by looking at it, you cannot see the difference. And so when I subcultivated it, I just took one of the larger ones because it was easier to pick off, and assuming that it was bacteria, right? And under the microscope, it's quite clear that it's not. Okay, and I know it's a yeast because simply by the size and also by the way it looks, bacteria look different. Yeah? So if you want to know which type it is, well, then that's of course a little bit more challenging. Then you have to do more investigations here. So, but I'm going to now take some of the E. coli here. Yeah. And that actually is a little bit, I have to tell you the following. That's, I have to tell you an anecdote. It is not fun. This was kind of told to us as a little bit of a warning when I was still working at university. They really said you got to be super careful with contaminants, because actually what happened is that this was kind of the story that was told in the lab, that another student who worked for months on a thesis essentially accidentally picked off a contaminant and continued to work for months with a contaminant. Right, he was not able to get any proper results, and only discovered this, I don't know, months later. Right, so it was essentially months of wasted time. Yeah? So you have to be really careful that you essentially know that what you're working with. Yeah, and you heat up this metallic thing. It's called an inoculation loop. Yes, and put it on the plate to cool. Can it cause to evaporate air, spores, bacteria, and inhale them? No, and I will tell you why. Because I'm going to cool it there where there are no colonies. Okay, number one. Number two, those bacteria because are quite moist and therefore sticky. Spores are or can be indeed a problem for fungi. Okay, these basically, that's indeed a case here. But for bacteria, even though some bacteria are able to form spores, and because the whole thing is moist, it tends to stick. Okay, so what I'm doing right now is I'm going to cool it. Let's do it over here. I'm going to cool it now here on the side where there are no bacteria. Okay, you can actually hear it. Huh? And I go over here. I grab again a small amount, and I dip it in the water here. I dissolve it a little bit. There are some questions arriving, I just see. Thank you very much. Maybe I already answered some of those. So be careful, no paper next to the fire. And then of course I have to kill off everything again. Okay, and we're ready to go again. So what I'm going to do now is I'm also going to take a little bit off from here and to put it on the side, maybe for drying it. This is way too much here. Maybe I can, we're gonna see if maybe I'm able to stain it as well. So let's put this away here. This is the one over here. And over here I'm going to put a cover glass on top. And yep. And these now should be E. coli bacteria. I'm also kind of waiting for a surprise now. Okay, so I put this in here. I know I have to change over to the microscope view. It's too bright. Yeah, I lost focus. Just a second. Let me see if I'm able to find it. So I'm not quite happy yet with the result, because this stuff that you see here, these are basically contaminants. Okay. Not contaminants, but these are, I don't know, those large things that you see, these are not, I think the density is way too low. Should have maybe picked up more. I'm not quite happy with the result here, honestly. And what you see, those large things that you see, these are not, I might not have picked up enough. These are not bacteria, okay? I don't know what this is. This is some kind of dirt on the slide. But these are not bacteria. So I'm going to do the following. I'm going to go over to phase contrast. Otherwise, I'm going to try it again. Yeah, not enough. No, I'm not happy at all. What you see over here, those rod-shaped structures that you see, these can be indeed bacteria, but I'm not happy. Yeah, those long ones, right? maybe some of those dots that you see here in the background, but honestly, I'm not happy with this at all. Maybe, I don't know if I got it out of focus, maybe. Maybe I'm focusing wrong. I don't know, just a second. Ah, here we go. That was it. That's here we go now. Ta-da! Yes, I got it. I got it. I think I was just focusing on the surface of the, of the cover glass. These here are, may I present, E. coli bacteria. Okay? These are basically rod-shaped, bacteria over here, and this is now in phase contrast. Okay, you see now quite clearly the difference between the bacteria and the yeast, because those bacteria are much smaller at a given magnification, and also they basically look much simpler. Yeah. So let's go up with the magnification, so away from phase contrast. So that's again back in DIC. This is now bright field. You see, that's in bright field, a little bit difficult to see now in bright field. And let's go up 60 times. Yes, I need to always adjust the brightness a little bit. And these are now E. coli bacteria, nicely visible. Okay. You might see that some of them, actually most of them don't move because they're connected. They stick to the surface of the glass, and some of them seem to be kind of jittering and moving around. That is because of Brownian motion. So very small particles, they under the microscope, like also microscopic oil and fat droplets and so on, tiny particles, they tend to move and randomly. So actually, maybe in that sense, this little accident, I don't know, I don't want to call it accident, but I kind of, the fact that the other thing was yeast and these are bacteria actually kind of, maybe is from an educational perspective quite good, because we can now see the differences between those. Yeah. So I'm quickly, check. Okay, what we have here is the following. What I would like to do, I'm going to wait a few more minutes because the slide with the bacteria with E. coli is now about to dry. Maybe in a couple of minutes. I don't have my coffee warmer here right now. What I'm going to do now is the following. I'm going to improvise now. What I would like to do is I would like to heat fix the bacteria. Okay, so basically these are the bacteria now that are put on a separate slide, and by heat fixing, Zoltan is asking a question here, which I'll respond. And by heat fixing, you will bake the bacteria to the slide so that you can stain them. I would like to do that, but I have to wait until it basically is air dried. And only then can I heat fix it. However, I'm going to do now something, I'm going to try to speed it up, okay? And I'm going to do this by carefully warming it, not making it hot. It's really important. As long as there's liquid on here, it should not become too hot. I'm just warming it a little bit to speed up the evaporation a little. Okay, yeah, it does speed up a little bit. Okay, so, yeah. Impatience is never good. If I were to heat it up too much, then basically what's going to happen is the bacteria are going to break open, and then then that's not what I want. I want to keep the bacteria intact, but I want to make sure that, so normally you have to air dry it, and this is now basically almost completely dry. Yep. I'm waiting until it's completely dry. So yes. So now it's air dried, and now I'm going to heat fix it. And this basically means is I'm going to pull it through the flame, and I'm going to make it quite hot, but not to the extent it should not start to smoke. I don't want to burn it, and you should just be able to hold it in your hand. And by doing that, the bacteria become baked to the glass slide, and they stick to the glass slide, and that's important because I would like to add a little stain to it. Okay, so I'm going to pull it through here once. Okay, yep, and maybe a second time. It's pretty hot, good, but yeah, good. So this is the bacteria have now been heat fixed. So they are now basically like egg in a frying pan. So the proteins in the bacteria, they form cross links with the glass slide. Yeah, and basically, yeah, what I can do now is I can add a stain. Okay, I have to wait, of course, until it's cooled down again because I've got this plastic surface that I don't want to melt. Okay, so that's fine now. So, and now I'm going to add a very strong stain. There are different ones you can choose, but yeah, in the past couple of months I've decided to use crystal violet because it's an extremely powerful stain. You directly drop it on here. You allow it to stain the cells for maybe a couple of seconds. And then what you have to do is you have to wash it off. How do you do that? By simply taking, yeah. So, yeah, there's a question again about how I made the agar medium. Okay, I will tell you quite in a second or so. Yeah, I think that's enough. And look what I'm doing next. I'm doing this, and not directly on it, but over it. Yeah, so I'm not directly doing this, but basically allow the water to run over bacteria. Okay, cool. That's actually, I think it worked quite nicely. So, I don't know if you're able to see that. Yeah, but it's stained, right? And by heat fixing it, essentially the bacteria cannot be washed away. So that is actually already a sign that this has worked quite nicely. And I need to simply dry everything here. And yeah, I'm just going to remove. Normally you have to do now the following. You have to air dry it again. What I'm going to do is, again, because I'm very impatient, I'm not rubbing it, but I'm just carefully tapping it like this. Okay. Yeah, and then you have to again wait until it's completely dry. So this is improvised, okay? Normally you don't do that, okay? But to save a little bit of time, you want to make sure that yeah, all the water is gone, because I want to use again oil immersion on this here. Yep. Okay. So that's basically how the slide looks like. You see, it's still purple here. It's stained. So let's have a look. Okay. I need to remove again the slide here. I'm going to now put the other one, okay, back in here. I'm going to go bright field. Okay, so this is now bright field, and I think, yeah, you can see quite nicely that the bacteria are now extremely well visible. 60 times. Of course, they're not moving anymore, right? Because they have been heat fixed and stained, but they're quite nice and dark. And this is now bright field, okay? Yeah, I think we want to also go oil immersion. So I'm going to add directly, and that's something that's a little bit different now. I'm not using a cover glass. I'm putting the oil directly on the bacteria. Okay. So yeah, first of all, without oil. You focus it. I focus it without oil. It's still, even without oil, it's actually pretty, how do you say, pretty good. I'm going to use again the focus lock lever so that to remember the position. No, I know why it worked so well. Because there was still a little bit of oil left from before. Okay, that's the reason. Okay, I was already surprised why the image was so clear. But there was, of course, some oil on the objective from before. And this is now, basically, these are now E. coli bacteria, which have been stained with crystal violet. Let's take again the arrow away. Yeah, and I think, yeah, I think we can be quite satisfied with the result. You can quite nicely see the, the shape, and sometimes there's still some over here. Yeah, it didn't quite dissolve yet, so there's some clusters here. And yeah, maybe some of them over here are just in the process of dividing, because what happens is they become longer, and when they have reached a certain length, then they're dividing in between here. Yeah. So they kind of extend in length and then basically divide. Yeah. Do you know any other application of crystal violet? Well, it's used in gram staining, so it is used in staining. I tell you the following. I've been doing a little bit of research because I kind of worried, where did all of the folks back in the 19th century and so on, how did they all discover all of those microscopic stains? And what I heard, or read, not heard, I read is that at that time, they were simply testing all sorts of stains that they could find, and they were actually talking to companies that were staining clothing. Clothing manufacturers, and basically they kind of tested them whether they're suitable for staining also other organisms, like bacteria and so on. So that's basically how they discovered the staining. Is simply they used the stains that were already available at that time in the clothing manufacturing process. So that's apparently how this was all discovered. Yeah, but you see that actually I'm quite happy. I'm fairly happy with the result here. Okay. Yeah. So there is now a question here, and maybe I'm going to now use the next couple of minutes a little bit to answer some of your questions. What did you use for making your own agar medium? Buying it is very expensive. No, it wasn't, because I did it the cheap way. What I've done is the following. I used for making those plates. Those plates here I bought finished, but those that are made, I simply got myself agar, which you can buy because it's used also for cooking, for making jams and so on. So it's not laboratory grade stuff that I have here. And I simply also have tried, I added some yeast extract, which I also obtained, and also something that I tried before, because I didn't have pure yeast extract. I simply took some yeast and boiled it until everything was dead. And then I kind of allowed it to settle so that the cells are gone. And I just took the liquid and basically used that as a nutrient medium. So essentially, there are different ways on how you can do that. And back in the 19th century, Robert Koch, the famous German doctor and microbiologist, well, they did not have those agar plates yet. So he used potato slices for actually growing bacteria. So that's essentially an interesting thing. But before I just realized one thing. I actually did want to show you something as well that has been requested, and that is the following. I want to quickly show you also the so-called the hanging drop method, which also Robert Koch invented. And I simply wanted to show it to you because indeed this was also somebody that somebody once requested, and I just wanted to show this simply for the sake of completion as well. It's called the hanging drop method, and in order, what you need is those slides that have basically are a little bit concave and that have this indentation. Okay, so they're flat on one side and they basically go in on the other side. And basically this is a technique that allows you to keep the sample alive for a very long time. Because if you make a normal microscope slide, then it's going to evaporate, of course, and the sample is going to dry out. But if you want to observe microorganisms over a longer time, because essentially you want to see how they divide, you must make sure that water does not evaporate. Okay, so Robert Koch at that time, he invented this method, and quickly, just for the sake of demonstration, I would like to show this to you. It's actually quite simple. So I'm going to again use then the E. coli bacteria. Okay, here we go. Okay, so I need again a little bit of water. I'm going to use a little bit more maybe this time. Okay. So this one is just a regular slide. It's not the concave one. This one over here is the concave one. And yeah, you do the same thing. Because I do not want to add any contamination into this plate here. Okay. You take again a small sample. Oops, that was a lot. You suspend it. If it's a little bit cloudy, then you actually know that the bacteria there. It is also accidentally had a little bit of agar medium as well. So, and now what you do is the following. Now you need a cover glass. This is now where things become a little bit, how do you say, you need a steady hand. And you need a little bit of Vaseline. Yeah, so here's the cover glass. So that's basically the suspension with the bacteria. Let's put everything away here. Just going to drop this on here. And what I'm going to do now is the following, I'm now going to add a very, very small drop of the bacterial suspension directly on the cover glass. And that's it. Yeah, so you see over here, maybe it should be very flat, right? So maybe I'm going to just also spread it a little bit. Okay. So I'm going to show you now the hanging drop method, and I want to observe the bacteria in this drop now for a longer period of time. And then I'm going to put this away here. And what you now need to do is the following. You now need to, so the drop is on the surface here, and over here you now need to take a little bit of, you cannot see this because it's a little bit overexposed, but that's Vaseline. Okay. It's Vaseline, and you now basically need to cover the side over here with Vaseline. I'm going to do this very quickly now, okay, just for the sake of demonstration. Okay, and I wipe my finger. You then take the cover glass with the drop on it, you flip it around, the drop is now hanging down, and I dropped it. Just a second, but luckily I dropped it. It doesn't matter. Okay, unfortunately, I don't know which side. Okay, yeah, but the drop is still there. Okay, a little bit. The drop is now hanging, and you basically put it on the slide, and you press down. Basically, what you now have is you have now you have it now sealed off. This means water is not able to evaporate, and now you're able to observe, hopefully, the bacteria in this drop. It's called the hanging drop method. And actually, Robert Koch, back in the 19th century, he was able to observe how bacteria divide in here. Yeah? So let's have a look. Okay. So I'm going to lower this here again, the microscope slide, and over here, the scope view. Yeah, cover slip always. Yeah. So, and here we go again. Just a second. Yeah, of course, finding the focus now is going to be again a challenge. Let me quickly have a look. I think we always should start at a low magnification. Yes, just a second. Give me a second time. Okay, I'm looking for some bacteria here. Maybe here somewhere, I don't know. I'm having some focusing issues. As always, just a second. Let's turn this down a little bit here so that you're also able to see something. And this here seems to be the drop. I need to kind of focus this a little bit. I'm a little bit upset because there is so much. Yeah, that's the Vaseline part here. Where is the drop? That's again one of those demonstrations. Let me find it. I'm a little bit upset now because I'm now stressed out because I cannot find the drop. See, that's one of those. But maybe this could be it. But because maybe I dropped the slide. Let me see. Yeah. Here we go. So I think here we go. Okay, I think I've got them finally. Bacteria difficult. So here they are, and here they are. Need to make it a little bit brighter. Okay, here we go again. Okay. Whew, okay, am I lucky? Yeah, focus on the edge of the drop, but I could not even find the edge of the drop right now. So, but in any case, what we have here right now are again those E. coli bacteria. And you see that, actually, this is actually a nice one over here. Yeah, some of them you can, and then basically what Robert Koch did is he added not water, but a nutrient medium, and then he could actually see how the bacteria were dividing. And because it was in a hanging drop, surrounded by Vaseline, the whole thing could not dry out. Yeah, so I just wanted to also share that with you a little bit. Okay. Yeah, a cheap way, draw a square on a regular slide with a Vaseline, put a drop cover slip and put upside down on the other side of the Vaseline edge, yeah. Another possibility on how you're able to find the focus more easily is by actually using a marker and actually drawing a small line on the surface, and then it's also much easier to focus. Okay, so... but I just wanted to show you this technique because it is of historic significance, yeah. So what I want to do now is wow, it's almost 1.5 hours again. I did receive again a couple of questions. Maybe I have already answered most of them. Yeah. How do we keep the Petri dishes clean from unwanted bacteria, from contaminations? Indeed, you only open. What I've done, I did not show this, but I wiped the whole surface with alcohol before I started the live stream. Number one. Number two, you have to actually use a gas burner to create this air stream so that bacteria and dust is not able to settle. And you should not keep the plates open too long. That's also, yeah. So there are several techniques. However, it can happen, and it does happen, that occasionally some contaminants will fall into the plate, and also they will make a colony. But very often those colonies look different. Either they have a different color, different size, or different texture, and then you know it's a contaminant. Okay, so just by looking at it, you sometimes can also see that. Yeah. Off-topic idea for a future stream. Yeah, the process of extracting lactose from milk. Okay, and then you kind of try to crystallize. I think that might be a little bit difficult if you're trying to extract it, because if there are any impurities in there, then it might be difficult to crystallize, but it would be interesting because there's of course lots of lactose in milk present, and recrystallizing and getting this out would be interesting. Yeah, I noticed that a lot of the organisms die after some time in the centrifuge. You mentioned you use it to concentrate water organisms. At which speed do you operate the centrifuge? This, first of all, this is not the fastest centrifuge. And generally, it is correct that if you have too many g-forces, then some of them might actually die off. However, I found that if you basically, I've even went up to 4,500 rotations per minute, and even then, many of them actually survived. I think the bacteria themselves, when they're small or single-celled microorganisms, they should not have such a big problem. I think the bigger problem are larger organisms, like I don't know, maybe some tiny worms or insect larvae and so on. But I think the smaller organisms should actually have less of an issue. Okay. Because don't forget, the smaller microorganisms, for them the surrounding water is a little bit like how do you call this, like honey. It's very viscous, yeah. And for the larger organisms, they are not quite as much supported by the surrounding water, and therefore I think for the larger organisms, yeah. But I would not be able to tell you a specific RPM that's suitable. Oh, I just realized you were not even able to read the questions. Sorry about that. Okay. Yeah. So here. Even at 100x, it seems like it's hard to look into the bacteria or any cell if it's not particularly large. So are they analyzed with other types of microscopes in research? That's a good point. So first of all, the thing is even if you were able to look into bacteria, because they are homogeneous on the inside. So there are no cell organelles inside bacteria, and for this reason, it's all uniform. So even if you were able to look into a bacterial cell, you would not really see a lot of structural details. So what do you do in order to analyze bacteria? There's actually three possibilities. Number one, you analyze the DNA. I think that's the gold standard. Number two, you can narrow it down by analyzing what the bacteria are made of, the chemical composition, lots of analytical chemistry. That's what I did. Or possibility number three, you analyze which nutrients a bacterium is able to use and which waste products it produces. So you see, so there are a variety of different possibilities on how you can narrow it down. However, the gold standard is nowadays DNA analysis. Yeah. So if you want to research them, yeah. No, I'm not involved in any academic research. How did you make the agar? I simply bought the agar in a shop or online. Not even how do you say? Not even laboratory grade. Yeah, and and sterilize the petri dish. I did not sterilize the petri dish myself because it was already sterile. Okay, that's the thing. I mean, that's actually a good point. So those plastic petri dishes, when they are actually delivered, they are already sterile, and you have to make the plates and pour the plates in sterile conditions. That's a little bit of work, and for this reason I bought also the ready-made ones. Yeah, would a candle also work? Honestly, the problem with candles is the soot, because the carbon. So this is an alcohol lamp that I have here. Honestly, would it work? Yeah. The question is, how well does it work? And it's not proper microbiological technique. And my recommendation is, please be careful with bacteria. That's all I just want to say. Okay, and that's why I've showed you the whole thing with the yogurt at the beginning, so that there is no need to actually grow your own bacteria, because there's simply a danger of contamination. Yeah, and then you don't know what you're growing. Yeah. How does the perifilm look like under the microscope, fully stretched? Okay, that's something you wanted to know. That's something we're going to find out. I've never put perifilm under the microscope. Okay, so we're going to give it a try. Okay, Who sells that parafilm? Honestly, I bought it also over, yeah, you can buy it in big rolls, but this one I also bought, honestly, buy all of my stuff on Amazon, yeah, because they've got pretty much everything that almost everything that you need. I don't know how it looks like. Okay. How does it look like? I don't even know if it's polarizing or not. Yeah. So let's stretch it. I don't know. Does it stick? Let's rip it off. So this is now stretched parafilm. And let's have a look. I need a lower magnification. Scope view again, microscope view, and yeah, neat. Ta-ta! I think it might be a little bit interesting. It does have an interesting texture. Let's go down with the magnification a little bit. Ah, look. It doesn't even. That's the IC. I'm gonna use just bright field. It doesn't even look that bad. Let's play around with the colors a little bit here. Yeah, it's actually quite. Okay, so that's a stretched parafilm. I have no idea who invented it. In any case, the parafilm is not only used for closing Petri dishes, but you can actually also put it over beakers, for example, for yeah, as a lid. Yeah, yeah. So yeah, looks like wood. Yeah. Yeah. So especially if something's stretched, indeed. I mean, tape is actually, I would say, more interesting. Regular sticky tape. Yeah. So let's quickly have a look at another question, maybe just very briefly. How do you close a burner like the one you have to prevent injury? I don't understand the question. I mean, close. I mean, this is an alcohol burner, and I simply put the lid on top, and then it does not have any more oxygen, and then the fire goes out. Yeah. So that's essentially. Yeah, I mean, alcohol flame is not that hot. Yeah, but you just do it like this, and then it's gone. Yeah. Genuine question: if one was to fart directly on a slide, would we find anything of interest, or likely just some moisture particles? Honest answer, you're probably not going to find a lot because it's methane and carbon dioxide. These are gases. And but having said that, in the intestine there are lots of bacteria. So I don't think that there are gonna be, yeah, in the air itself, it's not gonna contain a lot of bacteria. It's mostly gases, because the bacteria themselves actually they tend to be inside the intestine, and I mean. It's a little bit like, I mean, there are lots of bacteria in your mouth as well. And if you exhale, I mean, yeah, I mean, if you spit, for example, then of course there are bacteria there. Yeah? But I think it's actually, you need some liquid in order to spread those bacteria, so-called aerosol particles. Yeah? Like there are an infinite, off topic, like there is an infinite in the universe looking outwards, there must be an infinite trying to divide material inwards, I wonder. Okay, I will be honest with you. I think it was were the old Greeks that were wondering about that. How long can you divide something? Yeah, until they said they defined something called something that cannot be divided anymore, and basically something that cannot be divided anymore is called a atom. Tom means a means not, and tom comes from dividing or cutting, and so an atom cannot be divided anymore. And that's basically the limit. Yeah. So yeah, that's answers your question. Or you can go all the way down to the atoms, and then you are basically subatomic nucleus and electrons and so on. Yeah. So that's the thing. Yeah. So there is a lower limit. Okay. Unfortunately, my immersion oil has all solidified. Yes. This is often the case with natural immersion oils. No, you cannot melt it back. Okay, so you get yourself some new immersion oil, or you buy yourself synthetic immersion oil. And what makes immersion oil so special? The fact, the refractive index. That's it. Okay, so what I have here is the immersion oil that I have is from the company, directly from the company Olympus. That's the recommended oil. And yeah, it's totally clear. Many immersion oils that you can buy nowadays are actually yellowish. And these are natural immersion oils and they can solidify. This here is a synthetic immersion oil and it does not solidify. So I've had one bottle for, I don't know, almost 30 years now, and it did not turn solid. But the yellow ones that often come along with microscopes, they actually will turn solid, right? So that's the thing, yeah? So what I'm going to do now is the universe expanding or are we shrinking? Well, it's expanding. As of now, it's expanding. So that means, yeah, a lot of physics and hypothetical food for thought as well. What I would like to advertise, not advertise, but just for your info, in case you're new or so, I've now a couple of weeks ago moved the live stream to this channel over here. The main Microbehunter channel is now going to be a little bit more theoretical. I try to reorganize everything a little bit. So if you're interested in slightly more theoretical stuff, talking head explainer videos, please also have a look at my other channel. And this one over here will be more about practical microscopy and these live streams and so on. Okay, so let me quickly check here again if anything, if I overlooked something. Okay, yeah. Yes, I won't play around with bacteria, but it's very interesting to learn about this. Yeah, you can play. Honestly, I have no problem if you play around with yogurt. Okay, but otherwise, growing bacteria on agar plates where there's the danger of contaminants and so on, I'd be a little bit careful. Yeah, especially if you don't know what you're actually growing. Yeah, yeah. I think I'm going to about the yeast extract. I only find Vegemite spread online. Can I use that? I've never tried that. And very often those basically those spreads contain also salts maybe and flavors and so on. And self-made yeast extract performed not very well with E. coli, and I'll tell you why. Because my self-made yeast extract is not very clear. You see, it's very cloudy. And for this reason, I cannot see the colonies very well, like over here. Yeah, I mean, there's something growing there, but I cannot see it very well. And this is actually a disadvantage if you compare it, for example, to those, over here where, it's much more transparent and then you can see it much better, right? So, yeah, what I have done, however, in the past as well, I simply boiled some yeast, added some, what you do is you add 3 grams of agar, in 1 liter of water. So that is, no, mistake. It was 3 grams of yeast extract and 15 grams of agar, I think. Gee, I forgot. Yes, you make a 1.5 to 2% medium of agar, which means basically 15 grams of agar in 1 liter of water. You do the math if you want to calculate it down. And approximately 3 grams of nutrients. And basically I boiled it and then I was able to actually grow my own yeasts. So that as well, yeah. So the baker's yeast and so on, yeah. But again, yeah, you have to be careful. If you have other colonies growing in here, contaminants, you don't want to get that on your skin, okay? Because you don't know what it is. Yeah. So, but over here, all of the things here are basically from food, and that's why I don't have a problem with that because it's biosafety level 1, and we're kind of legal. In that sense, yeah. So I think I'm going to call it a day because it's already I've done a little bit longer, yeah. A baker's yeast, yes, yeah. Hope that you found it interesting, okay. Yeah. Amateur gram staining, yes, I can do that. Some months ago, I already did a little bit of gram staining, but that's indeed something that I can pick up again, okay. I wish you all the best for now. Hope to see you again soon, hopefully again next week. Okay, if something unexpected were to happen in the meantime that I'm not able to have the live stream, please yeah, have a look at the community posts. I'll keep you updated. Okay, enough for today. I'm already tired as well. It's time for me to go also to bed. I know that some of you basically have stayed up quite late at night as well, but I'll call it a day for today. I wish you all the best. Happy microbe hunting as always, and hope to see you again. Bye bye.